Happy Thursday, science students. Today we're looking at mitosis. We saw the beginning of reproduction the other day, looked at the basics of the cell cycle, sort of how the cell works, what its life looks like, and started looking at asexual reproduction, when there's one parent making two identical cells by the cell splitting. And that's what this is going to be today. We're going to look at mitosis and see what that actually means. First, let's do some quick questions here. When does a cell make a complete copy of its DNA? Why would a cell need to make another copy of its whole giant DNA molecule? That's typically only going to happen when it's getting ready to divide. It needs to make a double copy, a second copy of its DNA, because otherwise once it splits apart, where's the DNA going to go? We have to make sure there's two complete copies so that each baby cell ends up getting the exact same amount of instructions. Second question says, when a cell copies itself, how do the two daughter cells compare to each other and to the parent cell? Just like we saw in the last chapter with the DNA copying itself into two new molecules, same thing's going to be true with these cells. Each of the daughter cells is going to be identical to each other and identical to the parent cell that first created them. I think that visualizing chromosomes as this kind of tightly packed molecule of DNA, instead of being just all unwound as a big giant double helix, actually packed in tight into this more physical structure, is tough to picture, right? I mean, I'm seemingly seeing that on some of the forums answers I'm getting. That's an understandable reaction, I think, to this stuff. So I mentioned in the video the other day that there's a cool little animation. I had posted up in OneNote. I don't know if anyone actually bothered to look at that, but if not, I'm going to show it to you now on the next slide. So a minute and a half. I think it's just a good way to see it, where the DNA is a big molecule, kind of just keeps winding up over itself like a string that you just sort of keep twisting into tighter and tighter structure until it gets really, really tightly packed, condensed into a nice manageable form that we call the chromosome. So I'm going to show you that picture now. Then I'll show you a little actual photograph of a cell dividing. And that's what we're going to get into today.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope: chromosomes. Chromosomes are not always present. They form around the time cells divide, when the two copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
This is an actual series of photographs of cell division. The little bluish jumble right here in the middle, that's a pile of chromosomes. Just this little bunch of that structural compressed form of the DNA, and they're lining up in the middle of a parent cell. These little round yellow pieces right here are actually grabbing onto those chromosomes. So you can see these little kind of threads, little fibers coming off, and they're going to actually grab onto the chromosomes and start to pull apart now in opposite directions. So you can see as we move left to right through the picture series, these guys start to actually pull the chromosomes in each direction. The chromosomes now start to move to the left and move to the right, opposite sides of the cell. They get a little bit farther apart here. You can see this sort of fissure starting down the middle. And now here's where the cell starts to actually sort of pinch in right in the middle to form what's going to eventually be two separate cells. See it pinching in right there. This part's going that way. This part's going that way. We're getting what looks like two new nuclei, and the chromosomes are finding two new places to go, and you're going to actually get two brand new daughter cells compared to the original parent cell. So this is what mitosis is going to look a little bit like, and this is a type, again, of asexual reproduction. To refresh our memories from last time, why would a cell divide and make a copy of itself? Why would it do mitosis? In multicellular things, so larger organisms typically, this is to make more individual unit pieces of the larger organism, just more of our cells, more of the little building blocks that make up us. This can be for growth, which is really just adding more and more cells to increase the size of an organism, right? You're just making more of your own body cells. Eventually, you start to get larger and larger. It can be to replace or repair damaged or older cells, So we talked months ago about how bone marrow makes blood cells pretty continuously to keep your supply fresh and updated. Or in that vaccine video we watched, we saw the animated army of white blood cells start to grow really quickly by dividing just over and over and over again. Remember, we had the little like robot helmets and stuff on? You just get more and more of these cells all the time. That's all mitosis. Our body's doing mitosis all the time during our lives. One word we haven't really seen so far is right here. So this word somatic cells. So that's just showing that we call these body cells somatic cells, another word we use. Now we distinguish somatic cells from reproductive cells like egg and sperm. So basically anything that's not an egg or a sperm that we'll talk about next week counts as a somatic cell. It all counts as a body cell. You're really saying the reproductive cells, the sex cells, or the body cells. That's where we're going to kind of draw the line. They're egg and sperm are a little bit different. They have a different number of chromosomes because they need to then combine with each other to make a proper number. But everything else is going to count as a somatic cell. So all the ones we're looking at today are body cells that are somatic cells, just saying how do they reproduce. And one phrase you might see sometimes to remember this whole thing, how it works, is right here. Mito does mitosis, reminding you that body cells, again, copy themselves by mitosis, just to make more and more identical copies for whatever reason they're needed. Replacement, repair, growth, whatever. Now, if a single-celled unicellular organism uses mitosis to divide, that's just reproduction. Right? They don't have mates. They don't use sexual reproduction to reproduce. They don't have babies. But single-celled things will just make a complete copy of themselves, and boom, there it is. It's as if they had a kid. So that's still going to be identical reproduction, still going to be asexual reproduction, but two different forms of it, two different reasons really for it in different types of organisms. Here's where I want to slow things down for a second and give a visual definition to try and prevent too much confusion going forward. The word chromosome is used a little too interchangeably for my taste, so I want to try to clear up some things before we get into everything today. A chromosome, as we've seen, is this structure of condensed DNA. It's usually seen like this with sort of like a stick-like shape. It often has sort of like a pinch in the top here, where it's not a totally flat thing. It'll look a little bit like that. It says here that DNA is wrapped around protein, and in the video clip I just showed you, you saw that, where it's just sort of wrapped tightly. I didn't draw that here because it's really not important, but there is some protein there. The important thing is that the DNA all condensed and packed up tightly like this. Now, we showed the other day that we have 23 different types of chromosomes, but 2 of each type, one from mom and one from dad, for 46 total in every human body cell. The label here says this is chromosome 1. I just assigned a round number to it. That could be chromosome 17, it could be chromosome 22. It doesn't really make a difference. But this would be the copy of chromosome 1 we got from dad on the left, and the copy of chromosome 1 we got from mom on the right. And we call these homologous chromosomes. So we have 2 copies of the same type, but each parent's personal version of that chromosome. So all the same types of genes are on here. So maybe the eye color gene is right here on both chromosomes, but dad might give you a different type of eye color than mom does. You know, maybe hair color or height are down here. Your parents may have different heights, different hair colors, whatever, but the same type of gene is in the same place on each chromosome. That's the important thing to keep in mind there. Now, before a cell can divide, it needs to make a copy of every chromosome so that there's enough DNA to go around when the cell does eventually split in half. I always use the analogy of someone moving. So DNA is normally kind of a jumble in the nucleus. It has the double helical structure that we've seen in the past, but it's pretty loose and sort of all over the place, so that any recipe is accessible and can be read or copied while the cell is doing its work. It's not all wound up into a ball where you have to unravel it to get to the recipe. The DNA at that point looks like a big messy tangle of wires. So when the cell knows it's going to be dividing, it condenses the DNA and packs it into this chromosome shape, 46 smaller units in total, like getting all the wires and just winding them up into a more manageable size package. I mean, maybe take some computer or TV wires and put like a twist tie around them, some kind of zip tie, something like that. This way the cell knows where everything is, and it's a lot easier to move things around. A few chromosomes of DNA, nicely organized, are much easier to keep track of and ship out to the new daughter cells than miles of messy DNA threads just sort of going all over the place. If a cell just divides without duplicating its DNA first, then the new cells won't get the right amount of DNA. There's no way to know, well, did you send the right amount to this one and the right amount to this one? Maybe all of it mistakenly goes to one cell and the other cell gets nothing. So during the S phase of the cell cycle, the synthesis phase, when more DNA is made, the DNA in any nucleus is going to be fully copied. So now we see 2 copies of dad's chromosome number 1 over here. And right next to it, we see 2 identical copies of mom's chromosome number 1. And this gives us twice the normal amount of DNA for some small period of time. So we had the one version of chromosome 1 from dad, and that just got identically copied. So we have 2 copies of that right here. Same thing, we had one copy of mom's version of chromosome 1 here, and now we made 2 identical copies of that over here. Erase my ink here, let's see where it was. So the 2 duplicated copies of the same chromosome, both copies of chromosome 1 from dad and both copies of chromosome 1 from mom, hang out together and attach at their centers. And right now, when we do see this, we're going to refer to each one as a chromatid. That's going to be a sort of a different form of that word chromosome. So I would call this guy right here, this blue chromosome from dad, a chromosome, but I would call the 2 individual copies of it over here one chromatid and another chromatid, just to kind of distinguish that it's not the original form of it. It did change in some way. That's a word you'll see. I try to use it a lot, distinguish how I'm using it, but it can get a little tricky. And the little place where they're actually held together in the middle, right here, is called the centromere. A lot of times you'll see the centromere is the same color as the chromosomes, the chromatids, but I made it black here just so it stands out a little more. And it's at the center, like centromere. And I think of this typically as sort of like a little belt buckle. I drew this here with 2 identical chromatids lined up parallel to each other in sort of like an H shape. So like where these are, just straight lines going down. This one here would maybe have like the 2 parallel lines, and in the middle like an H. Same thing here, 2 parallel lines in the middle like an H. A lot of times you'll see it drawn in more of an X shape with the 2 copied chromatids actually laying sort of across each other with the centromere just right dead center where they meet. That's usually how you'll see it in pictures. I didn't do it here. I'll show you in a minute how that'll look. Here's where it gets a little tricky, unfortunately. Sometimes you hear this whole thing here referred to as one chromosome when it's actually these 2 pieces. If I am talking about this, I'm going to try my best to say that this is a chromosome and refer to each of these guys over here as chromatids, just so you know right off the bat every time I say these words what I'm talking about. This used to drive me nuts as a student. It was really confusing. I can remember sitting there in college studying for tests and trying to figure out what the heck the book was talking about, what my teachers were talking about, why they were using the word chromosome to refer to 2 totally different things. So I'm going to try to do this going forward to make it less confusing by referring to these single pieces here as chromosomes. And once it copies, and that there's 2 little versions of it, 2 copies of it here, I will really try my best to refer to each of these guys as chromatids, just so you have some sense of where we stand in the process. I can't promise it's always going to be that way. Some questions will do that. I may slip up and refer to it. You'll probably hear me later on in the video correcting myself as I say chromosomes for those. But we call these chromatids, and also you hear them sometimes called sister chromatids. I'll show you that on the next slide. But just try to keep that in mind as we go forward. Same kind of picture here. We see a pair of homologous chromosomes. Let's say this is the copy of chromosome maybe 14 that you got from dad, and the other copy of chromosome 14 that you got from mom. So each parent gives 1 copy of the same thing. 23 types of chromosomes times 2 gives us our 46 total. Now, before a cell divides, that DNA has to be copied so everybody gets the right amount of DNA. So now we see 2 identical copies of these chromosome 14 pairs over on the right side. So we have our homologous chromosomes. That's just saying we have 2 types of the same number chromosome. Again, 1 from mom, 1 from dad. Let's say this is dad and this is mom's. And then when they copy, the dads are going to copy. So you have now 2 copies of chromosome 14 here, and you have 2 copies of mom's version of chromosome 14 over here. Now, at this point, the copied chromosomes are kind of hanging out together. And where I showed you the more rectangular pieces, I just sort of drew them like this with a little sort of belt buckle in the middle. Normally, you're going to see them like this in more of an X shape. So this is actually showing that the 2 copies of the chromosomes actually kind of lie over each other. This is showing you 1 chromosome right here from dad, and this would be the 2nd chromosome in the other direction from dad. And this would be 1 chromosome from mom, and this would be the 2nd chromosome from mom. So it's not so much an H shape as it is an X shape. They're kind of crisscrossing each other rather than just sort of laying next to each other. But that's typically how you see it in that kind of X shape. And we call each one of these parts that goes together to make the X a chromatid. And together we call them sister chromatids, just to distinguish them that you're not saying it's a single chromosome over here. You're saying it's a pair of chromatids. This is really just a language issue. It's just a vocabulary thing, kind of a different way of saying these things. That's what I was saying a minute ago. It gets tricky just to kind of distinguish between these things. But we see these 2 sister chromatids hanging out together, and this round part right in the center here, that's the centromere. That's where they're actually going to attach to each other, and that's what's going to hold them together like a little belt buckle. I think of it that way. We'll see in a couple minutes how that's actually where the cell is going to pull from to pull. Maybe this guy goes this way, and this one goes this way when the chromosomes get separated, or the chromatids, get separated. One more version of this picture. A single unduplicated chromosome is on the left over here. So let's say this is chromosome 19 for mom. This is the mom version of the chromosome number 19. We'd also have a version from dad somewhere else. Doesn't really matter. And you can see here the genes. So we have all these little color bands here. That's the gene maybe for eye color, for hair color, for height, whatever. They probably wouldn't all be clustered like that, but each individual spot is a specific gene, a specific recipe for a different protein. Now, when the cell makes a copy of this chromosome, we see over here on the right now, 2 copies of the exact same thing, right? They look alike. We've got 1 copy of it right here, the second copy right there. This is now where we call it the sister chromatids. It's a double copy of the same thing. I know I just showed you how usually they sort of go like this and form more of an X shape. This is a lot more like an H shape like I showed you before, where they're kind of attached right here at this centromere, where normally you'll see them kind of like this in the X shape, like I'm drawing at the top right there. But this is how they hang out together. They sort of cross over and stay there. And then this is really good for organizational purposes, so nobody gets lost. So we always know exactly where each copy of the chromosome is, so that once we pull these things apart, we know one's going to one new cell, and the others go on the other new cell. These are the sister chromatids, just identical copies of the exact same chromosome, and somewhere else in the cell there'd be the exact same looking thing, but for dad's version of this chromosome. And then do the same thing for all 46 double chromosomes. With that out of the way, let's see how this is going to work. We'll start with one cell, the original parent cell. So there it is up top. And when that divides, it'll split into 2 and become 2 identical daughter cells. For that to happen, the cell parts will need to be pulled apart, as we saw in the picture. So these little things, these spindle fibers, these little strings basically, they're going to actually pull the cell parts away from each other. So they will line up on each side. So there'll be some projecting from right there, some coming from over here, and they'll actually just sort of grab onto all these little X-shaped chromosome pairs. And these guys will start to pull in that direction, and this one will start to pull in this direction. I love how that little fisherman does like that thing. But these are really just like that. They're like little fishermen just casting a fishing line out in the middle of the cytoplasm and pulling some chromosomes one direction, some chromosomes the other direction. And notice they're going to be attaching to the middle part. They're going to be attaching to the centromere. So those little belt buckles I was pointing out. The spindle fibers, the little lines, little threads from one side attach right there to that centromere, and the other guy on the other side, same thing, attached to that centromere. And they'll just pull this part of the X that way, and that part of the X that way, and these X things will start getting pulled here, and you end up with 1/2 of the X over there, 1/2 of the X over there. So actually I'm going to pull these sister chromatids apart to send one of the chromosomes one way, one of the chromosomes the other way. That's how you make sure that each new daughter cell is getting the exact same amount of information. All the little X pairs are getting split up and sent to either side of the cell. The spindle fiber is going to be made by the centrioles, the centrosome. That's one of those organelles we saw way in the beginning of the year that there really wasn't a lot to talk about. This is, really just basically what they do. They look like little pairs of Twizzlers or combo snacks, little logs that kind of go at right angles to each other. And this is it right here. That's that centriole right there. There'll be one that goes on one side, one on the other side, and they're just going to send out their little fibers, grab onto the centromeres, and pull those chromosomes apart in a little tug of war. One side gets some chromosomes, the other side gets an identical copy of chromosomes. This movement of chromosomes, 1/2 to either side of the cell, is what mitosis is. Mitosis is 4 steps and then one final part to finish the process. And the 4 letters that start these steps are going to be right here, PMAT, right at the top there. Those are the 4 steps, and cytokinesis is going to be kind of the extra part. I'm going to relate these letters to what's going on in each step. Notice as we start that we're looking at only one parent cell. That's asexual reproduction, right? You start with one parent cell. This is an interphase right now, which is how a cell usually is for most of its life cycle. And there are currently no chromosomes visible. DNA is just kind of a random tangle, as you can see there. The first step in mitosis is going to be called prophase, starting with a P, and P is also going to stand for pairs. That's how I want you to try to remember this. The DNA in a cell is normally just a jumble during interphase, but now when division is about to happen, the DNA has duplicated itself, made that second copy, and condensed down into the chromosome so we can actually see these pairs of the X's, the chromosomes. These are the sister chromatids we saw before. We can see 2 copies of this blue chromosome here, 2 copies of the red, 2 copies of it should have been more of a yellow color, but looks kind of orangey. But I've only drawn these 3 for simplicity's sake. This will happen for all 46 chromosomes in the cell. Also notice that there's no nucleus here. There's the cell membrane right here, but there's no nucleus that we can see because the nuclear membrane at this stage has just dissolved and the chromosomes are just sort of hanging out in the middle of the cell. This is also the first time in the cell cycle that the DNA in this shape of the chromosomes, these X's, is actually visible for us to see. In metaphase, the chromosome pairs line up and the spindle fibers grab onto the centromeres. And where do the chromosomes line up? They line up in the middle, which is also an M word. So that's your next letter to remember. M for metaphase, M for middle, and you'd have some centrioles, centrosomes over here. I'm going to send spindle fibers to these centromeres. Send spindle fibers right there, and this guy on the next step will pull this direction. This one will pull in this direction, And these pairs will start to separate out to individual chromosomes. Next, the centrioles, like I said, start to pull on the spindle fibers. So the chromosome pairs are pulled apart and move towards opposite sides of the cell. Where do they move again? They move apart with an A, and A stands for anaphase, which is our third stage of mitosis. In anaphase, we saw the chromosome pairs pulled apart, and one of each of these pairs moves over here. So we have 3 parts over here, 3 parts over here. They look identical to each other. And now part 4 is going to be called telophase, when a new nuclear membrane forms around each set of identical DNA. So basically you're getting now 2 nuclei forming within what is still one cell, because there's only one cell membrane there. T for 2 nuclei, T for telophase. The last part of mitosis is kind of a part of telophase, but sometimes some people actually count it as its own steps. I'm showing you here as a separate piece. Either way, it's called cytokinesis, and this is when the cell actually pinches in to divide the cytoplasm. So you see it here, the cell just kind of caves in on itself, the cytoplasm kind of pinches in, divides, and once this is complete, we have 2 brand new cells. So these are identical to each other and identical to the parent cell. These are 2 brand new daughter cells starting from our original parent cell that was up there. At this point, everything starts over, and you'll see now that both daughter cells are currently in interphase, just like they were originally as the parent cell. The DNA is back to kind of being a jumble. The chromosomes unravel. They open up again. The DNA just sort of unwinds into that familiar double helical shape, and the DNA is then available to be copied or used for whatever it needs to be used for. Hey, it's Kirby. Okay, let's do this. I don't know really what he's doing on here, but yeah. So here we got Kirby, and he's got his regular little Kirby features, whatever you want to call them, and his eyes and mouth, and then they duplicate here. I don't know why his cheeks didn't duplicate the same way. It looks like maybe they're acting as centrioles. Sure, whatever. So here we have prophase now. They've copied. We have the double copy right here, and then metaphase. They all line up in the center of his face. It's all line right down the center there. And anaphase, now they start to spread apart. So some go that way, some go this way. Telophase, you basically have 2 of them, but they're still connected. They 2 beginnings of separate cells, and now they finally split apart. And cytokinesis, we have 2 brand new happy little Kirbys at the bottom of the page. I don't know why this is so creepy looking, but this guy up here is just weird. It's kind of funny at the same time. I think this one's a little odder looking just because it's so very not correct looking. They're just way too far apart. At least with these ones, the eyes are kind of close enough together where it sort of looks like the original Kirby. I don't know. It's still really weird. This is pretty entertaining though, where he's just like lacking little feet in this middle one here. I don't know. I find this much more interesting than it probably ever should be. I also didn't make this. I want to make that abundantly clear. I don't know where I found this, but good job whoever made the little Kirby mitosis picture. Last thing. We talked about this a long time ago, but animal and plant cells don't divide in the same way. They can't, and that's based on how they're structured. The way we've seen it so far now is how animal cells work. They're able to divide easily because they only have a cell membrane, so 2 cells can be formed without any trouble. Why would plants need to divide a little bit differently, though? What do you think? Why would that have to not totally work exactly the same way animal cells do? Think about what plant cells look like. What do we always picture when we think of plant cells? Plant cells have a sturdy and rigid cell wall on their outsides, kind of outside and above their cell membrane, so they won't be able to pinch in to divide. Instead, a plant cell will do everything else the same, but in the last step of cytokinesis now, it starts to build a brand new cell wall down the middle. We call that a cell plate. This eventually will separate the 2 daughter cells that are being formed. The cell plate gets assembled in pieces, kind of like someone building a brick wall 1 stone at a time, until it's an entire wall. It's a very Cask of Amontillado style process. Oh, great reference, Mr. Tranchina. Thank you, voices in my head. In both cases here, though, the daughter cells are smaller than the original parent cell because it was divided in half. Each daughter cell then starts over and eventually grows to its normal size during the lifetime of that cell. That's it for today. Go complete the form in OneNote, and you've got some worksheets I put there also. That'll be due Monday. That's the 11th of May. It's crazy to say. We're that far along in the year. I'll have extra help tomorrow. That's Friday the 8th, I guess. Yep, the 8th. And again on Monday the 11th at noon on each day. All work for this week, all those worksheets and everything will be due Monday night, 11:59, so basically midnight. Let me know if you have any issues. Have a good weekend. Stay safe. I'll see you in a couple days.